Boa tarde a todos. É com uma satisfação que recebemos vocês para esse segundo webinar da série de comemoração dos 20 anos do programa Biota. Este é um webinar que será dedicado às pesquisas realizadas dentro do programa na área de microorganismos. Essa é uma área importantíssima em termos de biodiversidade, não só pela diversidade de espécies, de funções e de processos que envolvem micro-organismos, mas também do ponto de vista da importância que eles têm em grandes ciclagens e no funcionamento de ecossistemas. Se vocês tiverem perguntas, nós teremos a apresentação do professor João Setúbal, em seguida a apresentação da nossa convidada, a professora Jonna Mazet, da Universidade da Califórnia, Davis, e as perguntas podem ser enviadas para contato@biota.org.br. Good afternoon to all. It's a pleasure to welcome you all for this second round of webinars organized in commemoration to the 20 years of the Biota program. This second webinar is dedicated to microorganisms that have a very great importance, both in terms of diversity and also in terms of the processes they are involved. And it is a pleasure that we are going to have a presentation by Professor João Setúbal about the research that has been carried out within the Biota program over these 20 years. And after Professor João finishes his presentation, we have an enormous pleasure of having Professor Jonna Mazet from the University of California, Davis, and she's also the co-chair of a very large project in terms of microorganisms. This is the Virome Project, and she's going to tell us everything about it. So, without further delay, I will pass the microphone to Professor João Setúbal for his presentation. João, please go ahead.
Thank you, Jolie, for the introduction. I will start my presentation. Okay, so I will present a summary of results of funded projects by the program Biota Microorganisms, funded by FAPESP over these 20 years. And first, I'd like to introduce the subject itself, which is microorganisms. This is a very wide subject. And one justification for this kind of focus is that we live in a microbial world. Microbes are key players in many environments, and I have a short list here: in the soil, in the oceans, in the atmosphere, in agriculture, in animal health, in human health, and basically everywhere. And who are these microbes? I have another list here. So bacteria, of course, are part of the microbial world, but also archaea, fungi, and other microbial eukaryotes who go under the general name of protists. These are generally single-celled eukaryotes, in addition to fungi. And we should not forget, of course, viruses and bacteriophages. Everyone now, thanks to this pandemic, is painfully aware of the importance of viruses in our health, but viruses and bacteriophages are everywhere in large numbers and play also key roles in the environment. I also wanted to briefly go over certain key concepts for these projects. The first, of course, is diversity. We want to know who these microbes are, and behind the concept of diversity, there's the concept of phylogeny. We would like to understand their evolutionary histories. Of course, function is another key concept. What are these microbes doing in their various environments? And when we talk about function, we think about metabolism, and we also think about interactions. In interactions, by interactions, I mean interactions between microbes themselves, as well as with their hosts and other living beings in the environments where they live. And if you're talking about interactions, that means we're talking about networks. And again, behind the concept of networks, we have this general concept of ecology. And because we're talking about microbes, this means microbial ecology is a very important concept in any project under this general topic. And finally, we want to learn about all the microbes, what they're doing, their histories, but we also would like to apply this knowledge for the benefit of humankind. So this is applied science, and basically this translates into obtaining some tool, some biotechnology tool. And I'll talk more about that later. Okay, that was a 3-slide crash course in microbiology and microbial ecology. And I'd like to mention that even though the Biota program started in the year 2000, it was only in the year 2011 that FAPESP put out a call for proposals specifically directed to microorganisms. And I'll summarize here the key aspects of that call for proposals. One item was characterize microbial diversity in specific environments. The call also mentioned the creation of data banks of new microbial lineages. It mentioned as important aspects in proposals the aspects of ecology, systematics, and phylogeny. It also mentioned the question of what are the drivers of microbial diversity, biochemical investigations of microbial lineages, identification of microbial lineages with biotechnological and industrial potential, and the development of processes and products derived from these microbial lineages. So basically, those were the key points in that call for proposals. I will now present to you a summary of funded projects over these 20 years, over this span of 20 years. Starting with this summary table, FAPESP has different kinds of funding alternatives, and basically there are 3 represented here. First, there's the regular project, which is supposed to take 2 years, and there were 30 projects funded under this option. Another option is young investigator projects. These take 5 years, and there were 4 projects funded under this program of this kind. And finally, there's the thematic project. These are larger projects. They take 5 years, and 11 projects were funded under this option. So for a total of 45 projects. And in the next slides, what I will do is try to summarize what these 45 projects have achieved. Different dimensions were addressed in these various projects. And by dimensions, I mean the following. First, environments. Different environments were tackled were studied in these 45 projects. Some of them were surveys, microbial surveys of these environments, and others tried to assess environmental impact of human activities on the microbial populations living in these environments. Another dimension can be were the focus organisms. And then there was a diversity of aims, different projects focused on different aspects of the microbial world. In terms of environments, I have a summary list here. There were projects that focused on soil environments in the Amazon, trying to measure the impact of human activities in the Amazonian soil and also the Atlantic rainforest. There was a project that focused on composting, which of course is a microbial-driven process. There were projects that focused on plants, specifically Araucaria trees. These are gymnosperms. Mangroves, and they also address the impact of human activities. Estuarine sediments, specifically in the Santos, São Vicente area. There was a project dedicated to surveying the impact of human activities on river microbial populations. Another one focusing on howler monkeys, the microbiota of howler monkeys, comparing captive versus free-living howler monkeys. Another one focusing on the skin of amphibians, microbiota living on the skin of amphibians. And finally, a project that specifically targeted fungi, toxic fungi present in food products in Brazil. So you can see that there was a very diverse set of environments that were studied. In terms of organisms, many projects focused on bacteria and archaea, and specifically cyanobacteria, which play an important role in certain environments. Many projects focused on fungi, and there was one project that focused on amoebas. In terms of aims, there were diversity surveys, as I already mentioned. Projects whose main aim was to find out what roles do microbes have in their hosts. Again, as I already mentioned, several projects try to address the impact of human activities on specific microbial populations. There was one project whose main goal was education and extension as regards microorganisms. And then there were several projects that had as their main aims what I would classify as biotech-oriented research. And I will give more details about that in the next slide. These projects try to determine new biotech useful microbes and or microbe-derived products. And one of the topics that came up was antibiotic resistance. As many people now know, antibiotic resistance has become a major concern for human health because many pathogens have developed antibiotic resistance, and this is becoming a worldwide phenomenon of foremost concern. And it turns out that the microbial world itself can provide answers to these problems when you determine new antibiotic genes from microbial populations in diverse environments. Another topic that came up was looking for microbes that promote plant growth. also plant defense, and this falls under the general heading of biocontrol. Enzymes for biorefinery, polymer producing bacteria, enzymes for biomass degradation, which can help in various industrial processes, particularly biofuels, degradation of pesticides and other pollutants, and this falls under the heading of bioremediation, which was specifically mentioned in the call for proposals. And finally, there was one project that put together a natural products database, because without those databases, all the research that is done may be difficult to generate results that have impact on our daily lives. Now I will present a sample of publications that have come out from these various projects. And I need to stress that this is really a sample. It's not meant to be exhaustive, so I wouldn't have time to present to you publications from each of these 45 projects. So this is a sample, but I believe it's a representative sample. So starting with surveys, you have here a survey, the one that addressed the presence of toxic fungi in the Brazil nut supply chain. And that was a nice study that uncovered the presence of these toxic fungi in these nuts. On the right, you have a report about the microbial community structure in composting. And finally, at the bottom, you have a publication that addressed bacterial diversity in the estuarine sediments in Brazil. Moving on with surveys, this publication on the left is the one that addressed the presence of amoebas in a Brazilian river. On the bottom right, you have a publication that detailed results of cyanobacteria colonizing eucalyptus leaves. Eucalyptus, of course, is a major tree, major crop in Brazil. So understanding these kinds of populations is important to help deter pathogens. Environmental impact, I have a sample here of 3 publications. One is impact of silver nanoparticles in soil and aquatic organisms. On the right, you have microbial functional responses in mangrove soils. And at the bottom on the left, you have a publication that looked into how deforestation in Amazonia affects soil microbial communities. And this, of course, is a very hot topic nowadays. In terms of biodegradation, I selected these 2 publications. One that looked at marine-derived fungi and their biodegradation capabilities, and another one focusing on fungi, the biotransformation capabilities of these fungi, specifically related to this acid mentioned in the title. In terms of products, I point out these 2 publications. One is about pyrazines from bacteria and ants, and the other one is a tool to identify polymer-producing bacteria. I mentioned at the beginning that microbial populations play a role as in hosts where they live, and we have a nice publication here that looked at the symbiotic skin bacteria in frogs. And finally, in terms of resources, there is this publication here that describes this database of natural products. And these natural products are, as the title says, biological information from Brazilian biodiversity, most of it from microorganisms. So this finishes this very brief summary of results achieved by projects funded under this program. I'd like to now summarize what these projects have achieved. I think it's fair to say that the request for proposal expectations have been met. And very important, microbes are now first-class citizens in the Biota family. Initially, the Biota program was focusing on the macrobiota, animals and plants, but the FAPESP directors came to realize the importance of microorganisms in our biosphere, and thanks to that, we then had 9 years ago that call for proposals that I mentioned to you. And the third point is that viruses were notably absent from these funded projects. This is not to say that FAPESP did not fund viruses-related research. Of course it did, but it did not fund those projects under there wasn't a project funded under the umbrella of the Biota microorganisms program. I believe that moving forward, this is probably going to change. And we may get projects funded that are looking specifically into viruses and bacteriophages. And also, I should come back and say that it's very nice that we'll have a talk by Jonna Mazet right after mine, which will focus specifically on the virome. So I think that's a nice complement to the results that I have briefly summarized here. I'd like to mention now two recent developments concerning microbiomes. So microbiome is research that is specifically tied to DNA sequencing. And I realized that not necessarily all research related to microorganisms needs to be tied to sequencing. But because sequencing costs have gone down dramatically, it is my perception that microbiomes have become a central concept in any project or any collection of research that aims to unravel the roles of microorganisms in our environment. And as a proof of that, I will mention to you two documents. The first is this document here. It's an interagency strategic plan for microbiome research. This is coming from the US, and this plan was put together by the Microbiome Interagency Working Group, as you can see there. It was a document that came out a little more than 2 years ago, April 2018. It's a 40-page document. And what stands out when you read this document is that it's an effort to coordinate the various research funding agencies in the US, showing that it's important for these agencies to coordinate their funding efforts, because microbiome research is basically an interdisciplinary effort. And of course, I don't have time to go over those 40 pages here, so I will simply show you the three main points that this document outlines. The first is supporting interdisciplinary and collaborative research to enable predictive understanding of the function of microbes in diverse ecosystems. Second point is developing platform technologies. And the third point is to expand the microbiome workforce through educational opportunities, citizen science, and public engagement. So I think as FAPESP celebrates 20 years of research under the Biota program, I think it would be important to take into consideration the results of this workforce moving forward. And so that was for the US. And I have another document from Europe, where there's also a very large effort to draw attention to the importance of microbiome research. So this is a paper that was published in the journal Trends in Biotechnology, and it came out in December 2019, so less than a year ago. And the title, as you can see, is The Microbiome, a Life Science Opportunity for Our Society and Our Planet. And this is a nice summary of the opportunities that microbiome research offers in research and in innovation, which of course ties very well with the biotechnology initiatives that I mentioned previously in my talk. Various the projects that were funded by FAPESP can be seen under the light of biotechnology explorations. And I think this paper emphasizes the importance of these opportunities in funding research of this kind. So basically, I am at the end of my talk, and I'd like to acknowledge all the researchers who provided the information for the summary. My colleague Valeria, Maia, Merzel from UNICAMP helped put together this talk. Vera Sitting and FAPESP staff were the ones that helped with the logistics of this talk. Of course, Carlos Joly, who's the overall director of the Biota program at FAPESP, and of course, thanks to FAPESP for supporting the various projects that I described. And here you have the email addresses of Valeria and myself, in case anyone wants to contact us for any questions after this webinar. And with that, I would like to thank for your attention and turn back the microphone to you, Jolie. Thank you very much.
Well, thank you very much, João Setúbal. This was a very good presentation. I think you summarized very well the work that has been developed under the Biota program, focusing on microorganisms. And as you said, we didn't have any project looking at viruses, and that's one of the reasons why we at the steering committee of the Biota program thought that it would be very interesting to invite a researcher that's working with virus, and nobody better than Jonna Mazet, that is the co-chair of this big global project called Virome, and she's going to talk about using biodiversity science for pandemic prevention. Thank you very much for accepting our invitation, Jonna, and please go ahead.
Well, thanks for having me, and thanks for João's great introduction as well, because I hope to follow up on some of his points. Okay, So if we get the screen share going, I am going to talk to you about the Global Virome Project, but I want to just mention that my foundations are actually in biodiversity and conservation. So I'm incredibly proud to be able to join you and this esteemed group working in this area. I think one of the reasons that I'm working a lot now in the human health arena is because in working on biodiversity and conservation and recognizing that humans have the power, almost all of the power, to alter our environment, that it was critical that we really make some changes and address the human side as well as the animal side, even though I think I trained to be both a veterinarian and then a wildlife epidemiologist first with human cross training, but really so that I would have less to have to do with people. But my evolutionary process in my own lifetime scale has changed, and I do appreciate working at all levels. But it is this sixth mass extinction of plants and animals that we're in now that concerns me so much. And obviously our own species as well is at risk. The questions come in this arena: should some species be allowed to die out? Are we just managing for conservation dependent species and putting our resources there, when really what maybe we need to do is address the drivers that are affecting change? And that includes causing increase in climate problems, and that has species impacts as well as human impacts. The number of climate refugees around the world is growing and growing, but it also means that infectious diseases are changing, and I think we're all acutely aware of that and living with the tragic consequences. Bringing the One Health approach, or a collaborative, multi-sectoral, transdisciplinary approach to this problem, I think is critical. We need to be thinking along the lines of the intersections of human, animal, and environmental health. Originally, the One Health paradigm really took this three-pronged sort of paradigm, but I'm pleased to say now that we've been embracing even more the plant health side and pulling that out of the environmental piece as special, because of course we know our planet can't survive without the plants. We won't have air to breathe. We won't have food for all of the species. And so I think that's a critical and important development. If I take this One Health Venn diagram and start to think about the problems we could address, I'm going to talk to you mostly today about zoonotic disease emergence, or those diseases that pass between people and animals, or multi-species pathogens. But it's really the drivers behind them that are changing everything in all aspects of the programs that you're working on. It's land use, climate change, economic development, all of these pieces. Extraction is a major one, but underpinning that even is the societal influences, including policy and behavior. And those can be difficult or slower to change and find action on. And I think that's really what's happening for us today, is that while we knew a lot more about zoonotic diseases, we didn't know about SARS coronavirus 2, but we knew a lot more about coronaviruses in general and zoonotic diseases. It was the slower political will change that has really impacted our ability to respond quickly, unfortunately. And so it is time for a bigger paradigm shift. And I hope to convert, encourage, engage, and recruit some of you to participate with me in that bigger picture that I think we all understand why it's so important. We are living in this new reality. Here's the pandemic map. I think we all know your country and mine are faring very poorly in this response and control situation, but globally we have 22 million deaths now, 22 million cases now, and approaching, or maybe this morning, have met 800,000 documented deaths. I think if we had said a year ago that we were going to not be ready for something that could cause 800,000 deaths in the world, everyone would have laughed and been just incredulous about that, but it is the reality. And we knew it. Unfortunately, all of our modeling and predictions and documented recent history was showing that emerging infectious diseases, the majority of which are those that spill over from animals into people, were on the rise, and that we have been experiencing on the order of 3 new zoonotic spillover emerging infectious diseases each year in humans. And we don't recognize all of those. Obviously, not all of those show up as a big epidemic or a pandemic. We can document them, but they're not things that make it into the press always. But even the ones that do, look how many we have. It's on an exponential trajectory, and we need to get a handle and adjust that. And it's often the same things that we need to adjust for things like protecting our planetary health and all of the biota. We can't anticipate that we will be able to eliminate every spillover event or disease outbreak, but I'm here to tell you, we now know how to identify risks beforehand, which allows us to identify and stop them at their source. So hopefully the majority of what we can do is prevent these spillover events and epidemics and pandemics, but some will slip through. But if we're prepared, we have all that we need to respond, which includes detection. We can control them at their source rather than letting them run rampant through the world. Unfortunately, at this time, we only are aware or have been aware of about 240 zoonotic viruses known to make humans sick. And our past decade of research has shown that we are so understudying viruses. So I was happy to hear that they may become first-class citizens in the program as well, that we've so understudied them that we believe there are at least 500,000 out there that are available to spill over into us. Now, not all of those will make us sick, but we need to know about them. We need to understand the ones that can make us sick versus the ones that can't, so that we can start to rank better the risks from each one and understand how to control our behavior, change our behavior so that we reduce our risk, but also be ready and monitor those that are at the top of the risk paradigm. I'll talk to you a little bit more about that. So we already know how to do this, thanks to, again, some investments around the world, but the one I will speak to is the U.S. Agency for International Development's PREDICT project that I've had the honor of being the PI of for the last 11 years. It's a $200 million project, and we've worked in about 35 countries. But at the beginning, when USAID put out this challenge to preempt or combat the first stages of emergence of zoonotic disease that threaten public and animal health, that was sort of an audacious and crazy goal that most of us in the community said, wow, well, how would you do that? You don't even know what you're testing for, what you're looking for. We don't have the funding. And remember, this is 11 years ago, to do next-gen sequencing all over the world. How will we even approach this problem? But we dove in because it needed to be done, because there was the chance to make a contribution, but also the huge consequences of not trying. And this was, I think USAID, at least the US side, was the first to really put their money where their mouth was about the One Health approach. So we had been hearing for decades about the One Health approach and that it could be used, but really there were no large scale projects well-funded to be able to test that and to put it to work. So here's what we believed we needed to do. We needed to monitor, identify those viral threats that were sort of circulating in their evolutionary hosts, hopefully not making too many species or other populations sick. But that zoonotic pool of viruses, we wanted to identify them hopefully before or right at the point of spillover into domestic animals where they could amplify and then spread, or into humans directly. And so by doing that, obviously we need modeling, but the modeling requires good data so that we could stage and be ready for anything that was most likely to come. But we also needed early detection abilities. So we needed the laboratories to be prepared. And ideally, obviously, we're trying to lower those blue and red curves because most of the time we respond to epidemics and pandemics, frankly, way out beyond the benefits of control when the virus is burning itself out. And I'm sorry to say, in some places in my country, at least, we are having that exact experience now. So what did we do? Well, we engaged certainly first to bid to USAID. We built a consortium within the US. And then as the successful applicant, we were able and allowed and encouraged to engage out to countries that might participate. We used modeling, again, the best data available, which wasn't a ton, to identify where the science was saying we should look for the highest risk pathogens or potential pathogens, but that overlaid with USAID's portfolio. And it allowed us to interact with about 35 countries and bring together the ministries of health, environment, and agriculture so that we could do this concurrent sampling of wildlife, livestock, and people. The majority of our effort went into identifying viruses in the highest risk taxa that were identified by multiple modeling efforts, actually, which are rodents, non-human primates, and bats. But we were also able to look in sick people with fevers of unknown origin, working in the areas where there were those high-risk species interacting with people often, as well as healthy individuals that were working in high-risk exposure occupations and situations like guano harvesting or working in parks and interacting with species in pristine environments, or working with livestock and farm animals. And we worked with FAO in many countries, as well as the ministries of agriculture, to also look in the food animals. These are some of the high-risk interfaces where we worked. And I guess I show this to you because I want you to sort of have the awareness that the way that we are living on this planet is putting us into intimate contact with species, and more into species that we didn't interact with so frequently in our evolutionary history in most areas. Also, the inequities put certain parts of our populations at higher risk. And it's not glamorous. We're out there just swabbing, hundreds of people talking to communities, engaging with communities, getting their participation, and swabbing a lot of orifices of a lot of individuals, and people in the communities actually even providing their own swabs to us, going back into the back rooms of their houses. But it is interfaces like this. In the middle, I show you a bat guano harvester or tractant, a bat guano farmer, if you will, that bring bats to their property, harvest the guano, and put it directly onto crops that go to market. Some, like in this picture, do UV irradiation. We wanted to evaluate with them if they could keep themselves safe while harvesting and handling the fresh guano, if this UV would deactivate the viruses, and especially coronaviruses in this situation, before it went on to crops or before they took it to market to sell to other farmers. Oops. And what were we able to achieve? Well, we were able to achieve the proof of concept that we can take to scale a global virome project that would allow us to find those 500,000 viruses and identify the ones that need continuing monitoring, but also identify the ones that aren't making us sick. How did we do that? Well, we sampled more than 164,000 individuals, multiple samples per individual there. And while we were doing it, we trained almost 7,000 people and indoctrinated them into the One Health approach. We found about almost 1,000 viruses that were novel, and another couple of hundred of known zoonotics and other known viruses. That told us that we could, with a very inexpensive kind of lower tech platform, basically a traditional conventional PCR-based platform looking at conserved sequences of viral families rather than specific pathogens or viruses, we could find at the same time novel viruses in those viral families that have caused epidemics and pandemics in the past, as well as known. The knowns, let me tell you, were just as important because we were informing places and people and organizations and countries that they had viruses that they didn't even know they had in their country. We also were able to start to identify the host range of those viruses so that people could start to mitigate their risk. And maybe most importantly, I think we started a global immune system by training more than 60 labs in the 35 countries where we engaged to do this viral detection and discovery and characterization, which means not only can they continue to identify what is happening in their country, but they can be ready for that spillover event and identify viruses quickly and get them under control, hopefully much more quickly. And I have a bit of a proof of concept on that. This is just to show you we responded to more than 50 outbreaks, providing technical assistance to the governments where we worked, as well as sometimes responding in the field, actually doing the contact tracing or bringing the animal and environmental sampling to outbreak response. The majority of what we responded to were encephalitides and Ebola, frankly, in many countries. Also influenza on the animal side, looking at the animal die-offs and the early sentinels. And that helped inform us, especially for this COVID-19. It also helped us to continue to collect the data that showed that more and more deforestation, intensive agriculture, habitat degradation, and fragmentation are driving these emergence events. And it's the land use change, the act of changing it, that disrupts the ecosystem even more than what that change happens to be too, like from forest to urban or agriculture land. What it's becoming or where it's been is less important than actually disrupting the system. That's where we have these stress events that cause increased ability for transmission and contact. So what about COVID-19? How did we do? Well, we didn't know about COVID-19. We didn't know about SARS coronavirus 2, and we weren't working in the region of China where this virus appears to have spilled over and emerged. So we didn't find it. But we did find more than 100 new coronaviruses, and we waved the flag. We tried to raise the awareness that coronavirus were of big concern. And in some places, we got action, and in others we didn't. But we also proved that you can be ready for disease X and you can know more, especially, ooh, wow, that was odd. Hold on. Zoom did something very strange to the PowerPoint. I apologize. Okay, so one thing is that we had a handle on risk factors and hosts that help inform the global response. Around the world, our teams had actually stopped. The PREDICT project was aimed to stop at the end of September, and the teams were no longer paid or supported by PREDICT in the countries where we worked. At the end of that last September, it was 28 countries where we were still engaged. But those countries jumped into action rapidly. In China, our PREDICT supported lab and our PREDICT country coordinator was one of the first to identify SARS coronavirus 2 and actually provided within a week of getting sick patients, human patient samples, provided the sequence to the world to allow for testing to be developed, specific testing to be developed, even before she received samples from human patients and was able to jump in and help that way. Our teams in Thailand, Nepal, and other places in Southeast Asia and South Asia were jumping into action and using the PREDICT platform to identify the first cases that entered their countries and enabled their countries to put in public health actions, including contact tracing and quarantine right away. And I think that conveyed a much better outcome in many of the countries where we worked. We're actually doing an assessment now, looking at all the countries where we had engaged and comparing that to the outcomes of other countries. And of course, that's just a snapshot for our own, monitoring of what we were able to achieve. But even in places where the virus was coming later, in East Africa, for example, our teams were receiving samples from sick people coming into their countries from China, from Southeast Asia, and our labs were able to jump in and help to rule out that these people had SARS coronavirus 2, removing a burden of quarantine from those individuals, but also helping the country to know what was coming in and out. And now in all of these countries, you can see we're doing diagnostic support, technical assistance. Many of the places where we worked, our colleagues in the countries working with PREDICT were the first to publish on these kind of emerging viruses and coronaviruses. So they're providing critical technical assistance at the highest levels in their countries. And we could inform a lot more about the hosts because we had sampled so many potential hosts, especially for coronaviruses, especially bats. We were able to identify the most likely hosts for SARS-related coronaviruses, and especially this one. And for example, we found, as I mentioned, 100 new coronaviruses, but we found them thousands of times. And so we were able to start to get the host range. And for the SARS coronaviruses, it was really the rhinophilid and hipposiderid bats where we needed to look. But we had to be careful about how we messaged all of that information, because we don't just want to say this one bat in China is a culprit. We wanted people to understand that bats are widely distributed, and it's humans' behavior and our actions with the bats that actually facilitate the spillover, and that we need to protect bats. We need to protect biodiversity. Bats are the insectivorous bats control pests, the pollinators and seed dispersers connect fragmented landscapes, especially as we continue this land use change and degradation. We need the endangered plants to be able to connect to each other in their fragmented pockets. Also, we were able to message and need to continue to message that bats are important, but that if you wanted to kill them, let's say, you shouldn't, because those efforts have been generally unsuccessful, and it encourages recruitments of susceptibles, increasing transmission, because our project has shown when the bats are shedding the coronaviruses especially is right around the weaning of pups. So if you increase the birth rate, you're going to get more viral transmission opportunities. But really, it showed us that we need to do something differently. We need to take this kind of effort to scale and do it all over the world for everyone. And because we had done so much sampling and invested in all of the protocols and guides to set the foundation, we know now how to do it safely for us, safely for the hosts, but also we know how much it will cost and how long it should take if the world were to jump in collaboratively. So what do we need to do? We need to test every mammal species, and I would like to see even more than the mammals, but especially adding water birds first as the highest value target, and discover the viruses within them. That means sampling 1,000 to 2,000 individuals of each of these species in each ecoregion. It doesn't have to be every country. Small countries may not need to participate if they can't or don't have the time resources to participate, but we do need to cover the regions. As we continue to sample intensively across regions and within regions, we start to see the viral curve really saturating. And we know, because we've sampled at this level certain species already in the PREDICT project. We did not have the resources to do it for all species in the places we worked, but we were able to sample individual species, and we could see that we were saturating the curve and finding almost all, if not all, of the viruses in those individual species. That information then needs to inform strategies at the community level on how to reduce risk directly, but also how and what we need to do in the labs to be ready, how we can prepare systems for rapid engagement, including start to prepare and set up for sort of disease agnostic diagnostic platforms and even novel viral vaccine pipelines. So we need a global network. No one country is going to be able to support this for the world. No one group of scientists is going to be able to do that. We need to come together. And COVID has shown us that the scientists are willing and able to come together in amazing ways. We also need to have a data platform that allows us to share data, at least a minimum set of required data fields very quickly and easily. And then we need to use that to improve our risk ranking structure. We've started the risk ranking structure with an app that we're going to put out soon called Spillover. It's allowed us, because we wanted to just rank the risk from the pathogens or just the viruses that we are unaware of whether they're pathogens or not in the Predict project. But we didn't want to just use virology. That's been used in the past and hasn't shown to be adequate. We needed to use the host and environment circumstances. So we've identified about 40 risk factors that might influence spillover risk and then hopefully also inform on pandemic potential. And we enlisted help from scientists all over the world to help us weight how much each of those factors should contribute and then put together a risk ranking platform. Now, this kind of effort coming together across again, the globe and many, many countries participating is not cheap. To discover all the viruses, we believe it will cost us around $4 billion, but that is getting all the way out to that tail, as I mentioned. As you go further out on saturating the viral discovery curve, those last viruses are hardest to find. We could actually find the majority of those viruses that have zoonotic potential for about $1 billion. Now, when you think about the cost of this this pandemic on the order of $4 trillion US dollars, this is just a minuscule amount. But even if you think about the last SARS, we're talking about less than 10% of a single large outbreak like the last SARS to find all the viruses and start to be ready for them ahead of time. So that is what I'm soliciting your support and engagement. And Brazil actually has been very important, even in the foundations of the first idea of the Global Virome Project. Professor Carlos Merzel was one of the founders, if you will, with me in 2016, evaluating the potential for starting a Global Virome Project. And we are engaging with groups like the KAA initiative that wants to use autonomous drones to sample in the Amazon rainforest to find viruses. So we're very excited about the participation that is possible. I also believe that we need to strengthen the workforce in this One Health realm. I'm proud to be co-director now of a new huge project working with over 100 faculties in Southeast Asia and Africa to train the next generation of scientists to do this kind of work. And from here forward, I think even now in our own isolation situations, we can listen, learn, reflect, and act and get ready. We can commit to a transdisciplinary approach and begin to engage all the stakeholder communities. And I appreciate the opportunity to talk to you all about that today.
Well, Jonna, thank you very much for a very nice, though the time was very short, for presenting everything the Virome is doing, very provocative in terms of getting us from the Biota program more engaged with this initiative. And I'm sure that on our science plan for the next 10 years, virus will be one of the top priorities. We have ongoing, I think, 2 projects that started very recently, but they were looking on yellow fever, on the side of the impact it had on the monkeys population, not what happens to the humans, but what happens to the decrease in the population of monkeys in some areas. But they are just starting, so we still don't have any results. So now I will open for questions, and I've heard that Professor João Setúbal has a question himself. So João, it's with you.
Okay, thanks, Jolie, and thanks, Jonna, for a very nice presentation. I have two questions. The first one is that I found it incredibly interesting that you have been involved with this Global Virome Project for many years before this whole thing with COVID-19 started. So you were in the trenches. That's like it sounded to me. And I am curious to know what has changed from your perspective, from what was going on before the pandemic and now that the pandemic is here. In terms of, you know, perhaps once the pandemic started, more people started asking you questions, or the agencies started knocking on your door, or whatever.
Yeah, well, I will say this is my 4th international presentation this week. So that has changed. And in fact, I was doing one in India at midnight last night. So this is, my life has changed a lot from that perspective. But I think, I guess disappointingly, we were doing a lot of hard work in the trenches, as I mentioned, but also advocacy, trying to get elicit change and preparedness, and try to break down the silos of working pathogen by pathogen, and instead thinking more broadly about anything that might come and setting up systems to be resilient and responsive. That was disappointing. I've seen some strange things, like one of our TV news channels has put together this thing, and they have my face and a couple of other people, and they're showing clips of me talking about this in 2015, 16, 17, 18, and saying, you know, like the Cassandra, if you will, from mythology is had been telling us, and they were shunned or put aside. And that has happened, unfortunately, in yeah, it's all become very political and difficult, because some people don't want to say this was something we should have known about. I guess I don't think it's worth really engaging in the what ifs. It is now we should take advantage of this terrible tragedy to never let it happen again. It's much easier to talk to people now. We give the same kinds of talks, but people now are sitting home feeling the immense implications of what has happened. Some have lost loved ones. So it is an easier argument to make. I just hope that human nature, we can overcome our nature, because our nature is always to look at what just came. When I was working on the Ebola outbreak and Zika came, it was much of our resources were pulled from Ebola and switched to Zika, which is reasonable, except that you're still trying to find the answer to help the thing that happened before. I don't want to now be switching and saying now it's all about COVID, even though I'm actually an emerging threats and coronavirus researcher. I want to say, let's get ready for whatever might come. And many of what things that we need to do are the same for all of the pathogens. And but we need to understand and be ready and have that database, know the hosts, know the risk factors, know, you know, the receptor binding, all of that stuff that we can know if we study things and be prepared in advance.
Okay. I have another question, Julie. May I?
Yes.
Okay. Second question, last question is, you had this expression in one of your slides that we need to be proactive instead of reactive. And you also had the concept of spillover. And that got me thinking into this information that we have, which may be inaccurate, that a major source or the original source of this pandemic were the open air markets, wildlife markets in China. And that to me speaks to a cultural custom. And I was wondering, you know, the World Health Organization may do whatever it wants, and many agencies around the world may say this, may say that. Politicians may say all they want, but there's a cultural thing at the root of it. So how can you
yeah, I don't want to go on and on and on, so you have to give me you know, like we don't have time for this, but there are a couple of things to unpack there. One is the market that was originally kind of accused was probably not the initial spillover. Epidemiologically, it doesn't match with the appropriate time. It was definitely an amplification site where people were being exposed, but it could have been because they were exposed to a sick or multiple sick people in the market. We don't have epidemiologic evidence that that market was the initial spillover site, but it certainly was a place that multiple people converge on and got infected, but the timeline doesn't match for it being the initial spillover event. That being said, you are correct that the wildlife trade and live animal markets in general, because we've studied them now for a decade as part of the PREDICT project, and others have studied before that for SARS and Ebola and other things, are high risk interfaces. They are some of the highest risk interfaces for these spillover events, especially if the animals are live, because they are kept in high density, unusual, stressful circumstances, mixing with species they normally wouldn't mix with, including people in many circumstances, and being, you know, butchered and handled in often not at all biosecure circumstances. So those are big risks. We just had a paper come out on our Vietnam looking at wildlife farms and the wildlife value chain, all the way from hunting to butchering to actually coming to market, but also being used in live wildlife restaurants where you pick your animal. Now, we need to be careful when we say it's a cultural problem. I'm guessing many of you have picked a lobster in a restaurant, and it's not that different. So we need to be careful about our assumptions about whose traditions are stranger than whose, right? But certainly mammals in restaurants being selected and handled and butchered, that was the highest risk that we found when we were actually looking both serologically and at viral detection itself. So there are cultural problems, cultural issues. And again, I think the One Health approach, where we bring not just the scientists together, but the sociologists, the behavior change specialists, that's really what we need to attack this problem in the long run.
Thanks.
Okay, thank you, Jonna. Now I'm putting forward a question by one of the viewers. We have around 190 people watching us. So Fernando Maiello asks, with the rumors about a constructed virus around SARS-CoV-2, even already turned out as untrue, in your experience, you see DNA patterns that can differentiate natural occurring sequences from human-built ones?
Yeah, absolutely. And that's why I, amongst others, came out very strongly early on to say we don't believe that this was like the possibility was, it wasn't impossible, but it was almost impossible that this was a laboratory constructed virus, in that none of the markers that had been found in science. So you can't just build a virus without using some backbone, right? And the markers from all the known viruses were not aligning to show that this was something that could have been constructed by people. It has all the evolutionary markers, of a naturally evolving virus that hadn't been identified before. So you can absolutely, in this case it's RNA, but you can absolutely find those markers. You can also look at insertion points for how we know things can be modified in laboratories. We did not do any laboratory modification of viruses. And in fact, we worked with deactivated virus in all the labs that I mentioned. We do have, in some situations, a very tightly, locked down sample that could be available later for culture. But in all of those labs that I mentioned, the platform that we used deactivated virus so that there would be almost no chance of a laboratory accident.
Okay. Now we have a question by Valeria Merzel, who's my colleague at UNICAMP, and she's been helping João Setúbal to put together all the research that's been done before. Valeria, is with you.
Thank you, Jolie. Congratulations, Jonna, for your excellent talk and a great initiative. I think that one of the major challenges in this type of integrative and collaborative project is to get government authorities involved. So you need a lot of investments, you need a lot of strategies to put everybody together, working together. And what strategies are needed to be successful at the political level?
Thank you for that great question. And I think one of the important pieces is to think about the ethical, legal, and societal implications. So we certainly early on have engaged, frankly, in the PREDICT project, again, gives us a proof of concept. We engaged with governments and that's where we started with every country that we worked in. And frankly, even before this terrible pandemic, we didn't have a single government say, no, I don't want to participate. I've done a lot of international engagements throughout my career. I've never had one go so easily or have governments be so welcoming, because I think governments were aware that this was a tough problem that they would have to face and that they needed collaboration and support because nobody really knew how to do it. Thankfully, that's where a lot of our investment went into how to do it. And so that plus terrible circumstances make, at least for a short window in time, there to be a better opportunity. But we did actually, back even when I mentioned when we started thinking about this, we got amazing lawyers to jump in and to help think about things like the Nagoya Protocol, how we do benefit sharing for viruses that may generate vaccine or other opportunities. So we've worked hard to start that process, but these, I think, many of these laws and policies and international treaties are about to change dramatically too because of this. So we just have to maintain our flexibility and try to encourage openness, sharing, and sort of altruistic responsibility.
Thank you. One more question. Is it possible, Jolie?
Yeah, yes.
Did the project involve an educational approach for getting close to people and disseminating knowledge and the importance on the risks of crossing the border of wildlife and all the consequences? I mean, did you have any educational team to deal with this kind of issue?
Yeah, our funding wasn't dedicated to that, but we probably like the question, the people who are interested in this question, we believe that's essential. So we tried to modify and adjust and do a lot of things for free, frankly, to engage the communities. First of all, you can't come in PPE. Well, now you can, but then you couldn't come in white suits and sample people's food without people thinking you're nuts, maybe even coming after you, right? So we had to engage with communities from the very beginning. And really where we were most successful was where the communities became full partners with us and embraced, just like their highest level government where we started, we then went all the way from one end to the other, to the villages, to the community leaders, to community meetings, to really engage and say, what are you doing that you think might put you in contact with these viruses? How can we help you understand and then prevent additional contact? And in West Africa, after the Ebola outbreak, it was so moving to see that people really couldn't even say the word Ebola because their families often had been decimated by the disease. And by talking about it more from a perspective of let's understand it better for you so that you can protect yourself, so you don't have to wait for the white suits to come again next time and take your family members away. We heard from them, you say the word Ebola and protect ourselves. And we actually developed a Living Safely with Bats book. You can find it on our website, which is www.predict.global. And the Living Safely with Bats book is, we have it sort of culturally aligned with Africa and Southeast Asia right now, 2 versions, but it's more of a curated, narrated kind of picture book that could be used at all levels of communities with different, educational opportunities, and it's been translated into at least 12 languages now. So we're really kind of pleased with that, because again, we need the bats, but we need communities to be safe.
Jonna, I have a question myself. Actually, I have many questions, but it's not related directly with what you presented today, but it's related to the concept of One Health that is being. How this is being? I know that the aim is to introduce this in the next biodiversity COP as one of the alternatives of looking forward in terms of biodiversity vision for 2050, and that you all from the Institute have been engaged on that. And how are these talks going on? What are the reactions from different countries and positions? Do you think that is something that will be accepted as a concept?
Well, I think in some countries we saw movement before the pandemic. So actually China was starting and thinking about their own China Virome Project. And we're very willing to share data. I don't know what will happen now with all this political strife, but which is just devastating, honestly. But George Gao, again, like Carlos and I were of the first, I guess it was 18 of us that got together in 2016 to try and think about, and the outcome was we need to do this Global Virome Project. So he's the head of China CDC, if some of the listeners don't know, and has been, of course, under a lot of pressure and scrutiny during this whole pandemic. So I believe China will do their project. I hope that they will also support through their Belt and Road Initiative many other countries for engaging, and I hope that they will share the data, and I hope we can get back to some sort of global community in this realm. Thailand is another example. They were starting a pilot project for the Thai Virome Project. So those countries that were already ready to engage, I think will engage and move forward. But we're also hearing from a lot of powerful individuals, including the Trinity Challenge. Dame Sally Davies, who was the Minister of Health for the UK, has started the Trinity Challenge. She's put the Global Virome into the Trinity Challenge. Also, David Cameron has put into his pitch for the G20, the Global Virome Project. So I think there is momentum, maybe some better in other countries than my own right now, but I hope that we can turn that around.
Yeah, it's interesting. I was writing about the relation between the pandemics and deforestation and the impacts we may have from the Amazon. And there's a lot of people that believe that it's not true that the Amazon can be a big reservoir of virus. And indeed it is. I mean, if you start talking to researchers that are working in the Amazon, you always have reports on small villages where you had a problem caused by probably by virus, but it didn't become a big thing because the population is very small, so it doesn't make the headlines. It's known so by the people that are working there. And again, we also use a lot of wild animals. Is a tradition for in some regions that's very frequent, and it is the source of meat they have. We don't have this kind of market where you choose one animal. Usually the animals are already dead and you choose there, so you don't have this part of exposing people to too much. But it's curious, how do you see this? Is how big a threat could be hide in the Amazon region?
Yeah, I think globalization is going to put us all in contact, and that's what we're seeing. Certainly, we were working very early in the PREDICT project. We were working in the Brazilian and Bolivian and even Peruvian sides of the Amazon, and we worked with, especially in Bolivia and Peru, worked with indigenous communities. To just do just that. Politics kind of interfered, and USAID had to be pulled out of some of these countries, so we didn't get all the way to full conclusion. But I can tell you that where we've looked, what we understand is these spillovers are not infrequent. So these spillovers are happening. They just don't always have the perfect environmental and ecological circumstances to catch fire like this one did. And I think this is back to João, your point about the live animal market. It's when they catch fire, then we recognize them and they get diagnosed. But by doing the PREDICT work and I think the Global Virome work, we start to identify, for example, in South China, that people who worked with animals and went to markets were exposed, on the order of less than 1 to 3% of the time to SARS-related coronaviruses. And so this exposure is happening, they're becoming infected, they're mounting an immune response, but we're not seeing a lot of deaths or it catching fire. So we need to understand those circumstances that make that happen. We found the same with Ebolas and other filoviruses in Africa. We found up to, in some communities, 10% seroreactivity to very specific good serology, showing us that spillovers are happening, and the old dogma that if you have Ebola, the whole family or the whole village will die, it's just not true.
Okay. João and Valeria, do you have any other question?
No, I think I'm done.
You done? Okay, Jonna, so I thank you very much for participating in this webinar. I think it gives us enormous challenge of thinking if we could plan or could within the Biota program think about something like a similar Virome project in Brazil. I think there probably there will be support for that, and obviously it would be done in collaboration with what has been done before, and you and your team that have been running this large international project. So I thank you very much for bringing this discussion to us and participating in this webinar. And I hope keep in touch for the future when we are developing our new science plan. Thank you very much.
Yeah, thank you so much. Congratulations on your program. And I hope very much to be of support or to be working directly with you in the future. So thank you.
Okay. Bye-bye.
Bye.
And para todos os nossos participantes, muito obrigado. E terminamos aqui a nossa transmissão. Para os pesquisadores, coordenadores de projetos na área de micro-organismos do Biota, nós teremos uma reunião fechada daqui a alguns minutos. Entretanto, eu gostaria de lembrar a todos que no próximo dia 9 de setembro, daremos continuidade a essa série de webinars e teremos um que é dedicado à área de bioprospecção, que é justamente a identificação de moléculas ou de processos de espécies da flora, da fauna ou de micro-organismos que tenham um potencial interesse econômico, seja para a indústria de fármacos, para a indústria de defensivos agrícolas, para a alimentícia, ou mesmo para o uso como cosméticos. E eu espero contar com a participação de todos vocês novamente no dia 9. Vocês estão vendo na tela agora toda a lista de webinars que termina, na verdade, no dia 1º de dezembro, com uma transmissão mais festiva, uma comemoração realmente do 20º aniversário do programa Biota. Muito obrigado a todos, uma boa tarde.